So moving on to the next speaker, Dr. Angela Douglas, who is the Talcott and Elaine Cercaria Professor of Insect Physiology at Cornell University. Dr. Douglas is a graduate of Oxford and Aberdeen Universities and held numerous posts in the UK before moving across the pond. She is to Cornell, actually, where she is has her appointment. She is widely published and has written 3 books, including The Symbiotic Habit, so appropriate for her to lead our discussion of the origin and maintenance of host-microbe interactions.
Thank you very much indeed, and I'd very much like to thank the organizers for inviting me to contribute to this occasion. Can everyone hear me okay? Lovely. Okay. My talk today stems from my increasing appreciation that history, after all, really isn't bunk, and that our deep evolutionary history can provide us with valuable information about how our resident microorganisms influence our health and susceptibility to disease. With that in mind, I'm going to start off by taking 3 statements that you can treat as hypotheses or just conceptual notes. And my first statement is that our ancestors were multi-organismal before they were multicellular. And we have to bear in mind that we, the animals, are just a tiny offshoot of a tremendous evolutionary radiation of eukaryotes, most of which are unicellular. In fact, we're just down here. And our eukaryotic relatives interact all the time and time again, with microorganisms, especially with bacteria, in just the same way as humans and other animals do. And I'm just going to give you a couple of examples just to illustrate. The first one relates to the Microbiota, a group of bacteria that are increasingly being recognized as being important in relation to the human gut, members of the human gut microbiota and other animals. And these bacteria also associate with the surfaces of various of our eukaryotic relatives. I hope you can see the little dots, which are the Microbiota, on just the dorsal surface of this very complex unicell, Euclididium, which is a ciliate in the alveolates here. And in this location, these Microbiota contribute to the protection against natural enemies. Many of these bacteria associated with eukaryotes are found in cells. For example, many trypanosomes, not all, but many among the excavates down here, many trypanosomes contain bacteria within their cells, and these bacteria contribute vitally and absolutely required for their nutrition. With hindsight, I really think that we shouldn't have been surprised to find that our eukaryotic relatives act as a reservoir for various human pathogens. The Hartmanella, a reservoir, Hartmanella in the Neozoa, as a reservoir, for example, for Legionella. My second statement relates to just one particular branch point in this wonderful eukaryotic radiation, the one of most importance to us, which is, of course, the origin of animals. And this is a time when not only was there evolution of multicellularity, one of the several examples among the eukaryotes, but also a time of dramatic time of flowering, of origination of new genes. This is illustrated by some, I think, very important work of Thomas Domazet-Lozo and Dieter Taus, who calculated that some 16% of the genes in the human genome evolved at the time of the origin of the animal. That is, that these genes have homologs right across the animal kingdom, but not outside of the animal. And these are genes that originated at that time, are dominated, unsurprisingly, by genes associated with cell signaling and cell communication. This brings me to my second statement, which is that key elements in the animal signaling network clearly evolved in the context of pre-existing interactions with resident microbiota that had evolved and the genetic capacity to interact with resident microorganisms that were long evolved among our eukaryotic relatives. Now, Thomas and Dieter also noted that the genes that evolved at this time were a hotspot for disease-related genes. And we can, on the basis of that, it's not logically necessary, but of association, we'd anticipate some sort of interaction between dysfunction in cell signaling and communication associated with various diseases with perturbation of the microbiota. And I think, as Marty Blaser indicated right in our very opening talk, there's plenty of evidence that this is the case. And this brings me to my third statement, which is that the fundamental principles of the interactions between humans and our microbiota are conserved across animals. This isn't something that evolved especially in relation to humans. We're not just thinking about things that happened within the last tens to hundreds of thousands of years. We're talking about processes that are very, very ancient, evolved hundreds of millions of years ago. And one consequence of that is that if we're interested in the relationship between perturbation of the microbiota and human disease, we can make use of a variety of different animal models. Sorry, there we go. I'm not pointing in the right direction. And we have a variety of different animal models we can make use of. It's important to have a variety because, of course, different types of animal models are useful for different types of questions. So we have a variety of what we can consider as one microbe, one host models, and various many microbe, one host models. And what I'd like to do now is to tell you a little bit about some recent work that we've been doing on two model systems, and I'm going to start off with a many microbe, one host system, and then, secondly, consider a one microbe, one host system. So starting off with Drosophila. And I'm very grateful to the NIH who are providing support for our research. And the data I'm going to be presenting are predominantly work done by Adam Wong and Emma Ridley in the lab. And the basis of the work I'm describing is the ease with which we can generate germ-free or axenic flies. In fact, it costs the same amount as a tablespoon of domestic bleach to generate thousands of germ-free Drosophila, a little cheaper than mice, I think. So what we do is we just take eggs of Drosophila and we put them into bleach to decorionate, remove the eggshell, and the resultant embryo is completely bacteria-free. We carefully work with Wolbachia-free Drosophila, and we can transfer those embryos aseptically into sterile diet and let them grow up, generating our axenic flies. And as you can see here, there is no difference in the body weight of our flies, our conventional flies with the standard gut microbiota and the axenics, nor, importantly, any difference in their protein density. But there is a major difference in their energy storage. We find elevated levels of triglycerides. In fact, in this data set, 10-fold higher levels of triglycerides in axenic flies, females, relative to the conventional, and elevated glucose. And here, just to illustrate, is the abdomen of a conventional and an axenic fly, which we've eviscerated and stained with oil red. And I hope you can see there's a lot more lipid in the axenic fly. So when we remove the microorganisms from their flies, they have elevated lipid levels and elevated glucose levels. Very reminiscent of what Marty Blaser was mentioning earlier about the effects of antibiotics disrupting the microbiota. In our case, it was eliminating the microbiota. Although our data actually were very consistent with the data that Marty Blaser had got for the antibiotic-treated mammals, I was very struck that when they are the exact reverse of what we find for germ-free animals. And I think Marty mentioned that in response to one of the questions. And this actually brings up a general point about how we use models. I mentioned at the beginning, one of my statements, that we predict that fundamental principles of the interactions between humans and our microbiota are going to be highly conserved. I don't think, therefore, we should assume that everything is always exactly the same. We can quite routinely get rather different effects in different model systems, and that this variation can actually be very informative. Our working hypothesis as an explanation for this, and I'd be very glad for feedback for people who work on mammalian systems in relation to this, is that in the mammalian system, this effect of the germ-free mice being lean and being resistant to obesity is at least in part a consequence of the heightened energy harvesting by microbial fermentation in the hindgut. And the flies don't have that microbial fermentation. And therefore, actually, this system is rather better for studying the interactions with energy sensing and signaling because it's not masked by that microbial fermentation. That is a hypothesis, and I'd be very glad for feedback. As I mentioned a moment ago, we need to also consider the fact that the microbiota is variable, and there are a number of different microorganisms involved. The microbiota in these flies is much less diverse than that in mammals, and there are between 20 and 60 taxa, and it's dominated by 5 taxa, 2 species of Aphid-Buchnera and 3 species of Lactobacillus, particularly Aphid-Buchnera pomorum and Lactobacillus fructivorans in our laboratory culture. We've brought these bacteria into culture, and this means that we can take our axenic embryos and add what bacteria we want to them to generate notobiotic animals exactly as we design. And we have used that to explore further the effect of these bacteria on nutrition. And this is the work of Peter Neal. So what Peter has done has quantified the effect on glucose of the animal, which is, as before, elevated in the axenics relative to the conventional. When he added back in those main 5 bacteria, he found that they were, he rescued the conventional condition, and indeed all phenotypes that we've analyzed have been, of the host have been rescued by the big 5, indicating that low abundance bacteria do not have major effect. And it was also rescued by the Lactobacillus, but not by the Aphid-Buchnera. And indeed, we have found that to be common, that all the Lactobacillus and no Aphid-Buchnera reduce the glucose levels of these flies. He also analyzed the effect on triglycerides and got essentially the same pattern, with the one exception that I've reversed the order of the Lactobacillus and the Aphid-Buchnera. The Aphid-Buchnera reduced the lipid levels. In other words, we have microbial functions, and by function I mean impact on the host phenotype, which can be assigned to individual taxa, and that different microbial-dependent traits can be functionally independent. And this provides us with a tremendous opportunity to gain leverage to understand how different bacteria can interact with the nutritional biology of these model systems. I mentioned a moment ago about the importance that we shouldn't expect every system to operate in the same way, and this is illustrated by the difference between what I've just talked about and the other system we work on, which is on the P-aphid. And what we found, these are old data, there was no effect of eliminating the bacteria on lipid levels, the reverse of the Drosophila, and there was a significant reduction in protein content, which was accompanied by an increase in the free amino acid pool. In fact, of the 20 protein amino acids, all were elevated but 3, isoleucine, phenylalanine, and threonine, all of which are essential amino acids, contributing to our protein but not being able to be synthesized by animals. And we interpreted these data taken together that these 3 amino acids are provided by the bacteria, and their shortfall leads to a reduction in protein synthesis and an accumulation of non-limiting amino acids in the free amino acid pool. A very simplistic idea, but actually it proved to be essentially right. We've subsequently been able to show that the bacteria, which are called Buchnera, it's a single bacterial type, synthesize all 10 essential amino acids these supplement the short supply in the diet. I just need to remind you, these bacteria are not in the gut, but are in large cells located in the body cavity of this insect, occupying a volume equivalent to our liver, but different from our liver in that their cytoplasm is packed full of these bacteria with a very small genome size. We have established there is dynamic flow of nutrients across the interface between these bacteria and the surrounding cytoplasm. In fact, 58 different compounds in all. We've done this through work funded by NSF, and particularly the work I'm talking about is in collaboration with Klaas van Wyk and Anton Polyakov, very talented proteomicists, and a MetNet consortium funded by BDSRC and an Anglo-French consortium funded by BDSRC and ANR. So the approach that we adopted here was to take the metabolism-related genes of these bacteria, and from that infer the number of reactions and metabolites, and on the basis of that, reconstruct the metabolic network, and then apply flux balance analysis, which enabled us to quantify the distribution of flux through this metabolic network. From that, we generated what the inputs to the network are and what the outputs are. And the inputs were those 33 metabolites going into the Buchner. In other words, we inferred it entirely in silico. Here are the range of carbon and nitrogen sources that these bacteria were inferred from this modeling to require. Notice they're highly fastidious and inflexible, requiring multiple different carbon and nitrogen sources, completely different from their relatives, E. coli. And thankfully, our proteomic analysis demonstrated that the bacteriocyte, the host cell, is enriched in the enzymes mediating precisely this set of inputs, providing evidence that this isn't just sort of some in silico dream and that it has some empirical reality. We had transfer in the other direction of a total of 25 different metabolites, and we also noticed that several of the amino acid biosynthetic pathways in these bacteria were incomplete. So on the basis of that, we investigated how this could be, and I'm going to focus just on the missing enzyme in the final step in the synthesis of one of these amino acids, leucine. Now, this, the animal host also, and we all have this, an enzyme mediating the same capability. And we hypothesized that this enzyme was enriched in the bacteriocyte such that the final step was mediated by the animal host. The bacteriocyte proteome confirmed that this particular enzyme is enriched in the bacteriocyte, and Sophie Bouvain further confirmed this by raising antiserum against the aphid BCAT and showing that it is in the enriched in the bacteriocyte relative to the whole body. Furthermore, Callum Russell took the isolated Buchnera cells and showed that they released leucine at linear rates, and that the rate of production of leucine was dramatically increased by adding the bacteriocyte fraction, which contains BCAT enzyme, and this was eliminated by heat treatment. These isolated Buchnera, we can't get them completely free of BCAT, suggesting that that's the reason why we had some production of this leucine. And when we replaced this bacteriocyte fraction by recombinant BCAT, we were able to replicate the effect. Essentially, by this and other approaches, we now have evidence for shared metabolic pathways in the synthesis of 5 out of the 10 essential amino acids. And this is really a case of metabolic coevolution, with the loss of Buchnera genes coding reactions that are also present in the insects, and the dedicated expression of the compensatory host genes in the host cell. This is my last slide, and I want to make 3 general comments. The first, arising from the last thing I've been talking about, is that metabolite exchange between the animal cells and resident microbiota can be underpinned by co-evolved animal and microbial metabolic networks. We've shown this for this highly co-evolved simple association. I think this may be general to many more complex types of associations. Secondly, that the microbiota influences the metabolic configuration of the animal, and we hypothesize and are getting increasing evidence that they interact directly with the nutrient signaling network. And finally, a general point that the very architecture of animal physiological systems is predisposed to modulation by our microbial partners, simply because we were multi-organismal before we were multicellular. Thank you very much, everyone.
Thank you, Dr. Douglas. We have time for a couple of questions. Forum members. David?
Angela, it was a great talk. You mentioned that your work, the work you described with Drosophila was with Wolbachia-free.
Yes.
Have you looked at what might happen with Wolbachia?
We're getting some data through from our GWAS analysis that indicates that those strains with Wolbachia have heightened glycogen levels in response to elimination of the gut microbiota. We haven't followed it beyond that. The effects are not very wide-ranging, but there certainly are effects. So we think there's interactions between the Wolbachia and the gut microbiota.
And presumably it's not only just metabolic and direct that way, but indirect through innate immune.
Precisely. And in fact, this is closer to speculation than fact. We suspect that some of these metabolic effects are mediated through differences in the immune system. And we have evidence for immune-related effects as well. Yeah.
Go ahead, microphone.
Can you provide any thoughts as to whether you think at one point Buchnera might have had this complete metabolic capability, and if it lost genes involved in these critical pathways, do you think that was in response to a particular selective pressure or a random event?
So I skipped over some interesting biology here, that these bacteria are subject to genomic deterioration as a result of obligate vertical transmission with bottlenecking of their populations over more than 100 million years. And they have lost almost all of metabolism-related genes that are also present in the pre-existing in the animal host. And these shared metabolic pathways means that actually there is the transfer of intermediates between the two partners. Some of these pathways are actually quite complex as a consequence of host compensation for genomic deterioration of their microbial partner.
One more question.
David Mills, UC Davis. You presented a very bacterial-centric view of Drosophila, and you mentioned that the yeast are used for food. Even if they're just used for food, are they still having a metabolic impact? Different yeast act differently. I know that flies are covered both inside and out with yeast.
Yes, you're absolutely right. We work entirely with dead yeast, and the effect of the yeast on nutritional status of the fly is the same whether it's dead or alive. However, the yeast cell walls may well be very important in relation to the immune responsiveness of the insect. Now, there's a whole new other story there.
Thank you, Dr. Douglas. We'll have time at the end of the session.